So today is the last talk for this week in the context of the International Graduate School and the International Guided Tour that was organized. And so the last guest is Francesco Licausi. Francesco, you are from Italy. You did your PhD. No, sorry, you did your PhD in Germany, in Potsdam, and then you spent one more year, I think, in Germany. Then you moved back to Italy in Pisa. And since 5 years now, you have a permanent position in Oxford as a professor position, and you work on oxygen sensing mainly. And so we are really pleased to welcome you today. So, and to also for your work for the student, for our master student, because he gave also 3-hour courses, 3-hour roundtable, so a lot of work for him, and time for this seminar. So please, Francesco.
Thank you very much, Jean-Philippe, and thank you all for being here. I understand it's quite a long list of seminars that you're having this week. So it's a real pleasure for me to be here and appreciate the scientific community, the excellent students that are participating to this course and that are interacting with me. So for those who might have read the title I suggested initially, you might notice that I changed it slightly. Because of the discussion I had with some of you, I left the idea of perfecting perception or changing mechanisms of perception. But while initially I was planning to present our work more on the area of synthetic biology, where we interfere with what nature has provided us with, I rather decided to present work that is more connected on the study of evolution and diversifications of the mechanisms of oxygen sensing, which is another area in which my group is particularly focused at the moment. So let me start explaining why we study oxygen sensing. When I started as a master's student and I was suggested to do my thesis on anoxia and oxygen sensing, I was a bit puzzled because I thought this is quite irrelevant for plants when they actually make their own oxygen, right, through photosynthesis. Reality is that actually plants experience low oxygen conditions and their cells starve for oxygen more often than actually we think. So one of the most well-characterized conditions in which plants are exposed to hypoxia, or at least some of their organs, is flooding or waterlogging. And this is because in flooding conditions and in water, oxygen as a gas diffuses 10,000 times slower than in air, which then means that at least roots in this case will be consuming very rapidly the oxygen near them, and this will take some time to be replenished. If we consider then the fact that, yes, plants do photosynthesize, they can transfer through dedicated systems oxygen to the most remote district, like root tips. But if, for example, the water in which they are submerged is particularly muddy or during the night, then there is no way for oxygen to be produced. And so these tissues will experience real strict hypoxic conditions. And of course, this will impact plant physiology. And if we consider crops, this will limit their productivity, if not threatening the life of the plant. So this is how essentially the study of oxygen sensing was initiated in the early 40s. More and more evidence actually indicated that plant tissues and some organs actually experience low oxygen levels even in perfectly aerobic conditions in the environment. And this is not potentially a threat for the plant. It's part of their regular developmental program and their growth program. But it's caused essentially by the establishment of bulky tissues that are poorly vascularized, by the establishment of layers of cell wall modifications that are water impermeable, but also gas impermeable. So they limit diffusion. They are caused by very active mitochondrial respiration, and in very specific cases also by the production of oxygen-binding proteins that act as buffers to protect certain oxygen labile complexes, such as in nodules. So what happens when these tissues are experiencing hypoxia, either because of the environment or because of developmental programs? Bit of basic knowledge, and this is a slide I presented yesterday to the students. We all know that glycolysis can keep running and potentially produce ATP, but needs to have oxidized NAD being regenerated. And this process is typically assisted by fermentations. Plants potentially do both lactic fermentation and alcohol fermentation, but they actually prefer ethanol fermentations because lactic acid would tend to acidify the cytosol. Maybe a bit less known is that another way to regenerate oxidized NAD is through this cycle mediated by non-symbiotic phytoglobins, which essentially reoxygenate nitric oxide. So nitric oxide is produced under hypoxic conditions because nitrite is used as an alternative electron acceptor by cytochrome, given by cytochrome C oxidase. And when accumulated, this can be toxic for plant cells, so it needs to be scavenged. And phytoglobins carry out this function with the additional benefit to oxidize NADH. So here in red, I highlighted all the enzymes that are transcriptionally controlled to provide an accommodation of suitable metabolism to cope with hypoxic conditions. And this was more or less what was known when I started as a master's student. And the group I was in was strongly interested in understanding how the genes coding for these enzymes are regulated. And we focused, particularly my thesis, my master's thesis initially, and then my PhD thesis, focused on a group of ethylene-responsive factors which are induced under hypoxic conditions. We named them the hypoxia-inducible ERFs or HREs. Turned out that this wasn't the best class of transcription factors to focus, because actually the more important than these that are controlled at the transcriptional levels are these three here, which are instead constitutively expressed, if anything, under prolonged hypoxia, they're slightly repressed. But we will see they're actually controlled in another way and directly by oxygen levels. So something that characterizes all five of these transcription factors and is quite unique in them is the very conserved N-terminus, which, if you remember your favorite proteins, you might remember that probably this has conserved domains, but these are normally not at the N-terminus or at the C-terminus. And there is another weird thing about the N-terminus of these proteins here, that is that it has a cysteine in the second position. And proteins in plants, but also in our cells, dislike to have a cysteine in the second position. So it's very rare to find proteins with a cysteine in the second position. And this is because of a pathway that is meant for degradation of proteins that has been named, well, initially was named the N-Drude pathway, now has been renamed as the N-degron pathway. And essentially this dictates the stability of the proteins depending on the N-terminal amino acid that is exposed. This being exposed either because of the activity of methionine aminopeptidases, that would remove the starting methionine, or because of endoproteolysis, which would expose residues that are normally not found at the N-terminus. Here in my presentation, and for oxygen sensing, it's worth only focusing on these reactions here on the left, where maybe you can move and I can show you that actually the ERF7 are ideal candidates for being substrates for this pathway. So indeed, if this ERF7 are produced, then co-translationally the methionine is removed, and this exposes the cysteine to the activity of N-terminal cysteine oxidases, which in plants we call plant cysteine oxidases, or PCOs, which use molecular oxygen to label this protein for subsequent reactions that eventually polyubiquitinate the protein and send them to degradation by the proteasome. And I will be referring to these enzymes ATE and PRT6 and BIG later on, so maybe keep them in mind. ATE is an arginyl transferase that essentially adds an arginine post-translationally to the N-terminus of a protein to label it for degradation. So arginine is a typical signal for protein degradation. And something else to keep in mind is that nitric oxide is also important for this process, but we don't know exactly where this operates. We know that through a series of experiments that this is not occurring at the level of oxidation of the cysteine, which instead is purely enzymatically mediated, but we don't know if this is at the level of arginyl transferase or the ubiquitin ligase. So this means that the ERF7 were perfect candidates to be controlling the molecular response to hypoxia, and through a number of different experiments, our group, but also the group of Mike Holsworth in Nottingham, demonstrated that indeed they control most of the genes that I mentioned initially. And work in Bayreuth by the group led by Angelika Mustroff also identified the DNA element that is recognized by these transcription factors, which was named HRP, or a hypoxia responsive promoter element, which is slightly different from the regular most common GCC box that is recognized by the rest of the ERF transcription factors. So we use this element linked several times as a kind of reporter for low oxygen conditions when we fuse it with different reporter genes. So we can use it with luciferase to, for example, demonstrate its responsiveness and the fact that it's activated quite at relatively mild hypoxia, 10% oxygen. We use it when fused with GFP to identify new regions of plants that are characterized by low oxygen conditions. And by doing so, we actually observed that the shoot apical meristem is a very distinct hypoxic niche. And following this, we also observed that other tissues that contain cells that are highly proliferating are characterized by the same. So, for example, galls caused by Agrobacterium tumefaciens also reach very low oxygen levels, but also lateral primordia. Quite surprisingly, we don't see the same at the level of the primary root, which might indicate that the primary root meristem is not hypoxic, but might also indicate that it is a relatively insensitive, so maybe it's calibrated to a different oxygen tension. Keep in mind the fact that the shoot apical meristem is hypoxic, because I will bring this information back towards the end of my presentation. So I mentioned that the ERF7 transcription factors are key regulators of the hypoxic response, the response to hypoxia. You might wonder if this is the only protein which has this feature. And I told you initially that proteins are rare with cysteine at the N-terminal position. We have about 200 proteins in the whole Arabidopsis proteome. Two of them have been also characterized as substrates of the N-degron of this branch of the N-degron pathway. Both of them are transcriptional regulators. They are not exactly transcription factors like the ERF7. We have little zipper2, which is a microprotein, which arose essentially by deletion of portions of HZIP3 transcription factors, and is quite specifically expressed in the shoot apical meristem and controlled by the oxygen levels there to essentially dictate how many, or how rapidly new primordia should be produced. And the second substrate to be identified is vernalization 2, which is part of this complex that adds methylations on specific lysine residues on histones. And very recently, I think a couple of months ago, the group of Daniel Gibbs published the interaction of this hypoxia-regulated VERN2 in essentially creating the right landscape for P4 to regulate cell expansion rather than cell division in leaf primordia. So again, key message here is that the ERF7 are not the only substrates, but we have at least 2 more, and probably other groups will add a few more in the near future. So when typically I present this work, I often receive the question, which is, how well conserved is this pathway, especially when we compare it, for example, with oxygen sensing in animal cells, which is conserved in all metazoans. So this is a question that we asked ourselves, and we started working on this quite some time ago, when we considered 2 aspects of plant evolution. First, the fact that anatomies have changed and growth rates change across different plant species, also considering different divisions in the plant kingdom. But also that essentially plants diversified and evolved in times when oxygen levels quite dramatically changed. So it would be possible potentially to observe in some species sort of relics of maybe an older mechanism for oxygen sensing, and then address the question as to whether these ERF7 mediated responses are universal or rather maybe flowering plants specific. So to present the work, I would like to start indicating those who did the job. And this was work led by Laura, a postdoc first in my Holdsworth group in Nottingham, who then moved to my own group in Oxford 3 years ago. And another very important collaborator for this is Hans van Veen at the University of Groningen now, and who helped us with bioinformatic analysis. So what we did here was starting comparing transcriptomic responses to hypoxia in a range of different species that might correspond to different branches of the land plants in the plant kingdom. We ignored unicellular algae. This is a part that is actually studied by a colleague of mine in Oxford. So we looked at bryophytes, including, so we used mostly the most common model species for this, except for ferns, where we used the Pteris vittata, mainly because we had a colleague in Pisa who was using this for phytoremediation purposes, and so we had the plant available there. So we included then Marcantia polymorpha, Physcomitrium patens, Selaginella as a lycophyte, Equisetum, which was growing in Mike's garden at that time, and then these data for Arabidopsis and rice were already available in several data sets. So we ran a very simple experiment with exposing plants at 1% oxygen for 8 hours, a condition that we know doesn't kill the plant. And then we measured the level of the number and the level of induced and repressed genes in these 7 species. Of course, we observe quite a diversity of responses in terms of magnitude of response and in terms of a number of genes being affected. But comparing them, just looking at genes, is hard and unfair, because of course certain species underwent genome duplications, for example, or certain families might have expanded. So to analyze this data, we need to resort to the concept of orthogroups, which you might have heard of before. Essentially, orthogroups would be groups of genes from different species which have a last common ancestral species. And these groups would be originated from one single gene in the last common ancestor of the species considered. So they can be grouped in phylogenetic trees. So we then looked at orthogroups, and first we categorized them among orthogroups that are conserved in all land plants, or those that in a maybe flowering plant biased way, we named as gained or lost, depending on whether we found them, let's say, towards angiosperms or more towards bryophytes. And we also had, of course, a number of orthogroups that are more lineage specific. And when we looked at the number of genes, of orthogroups differentially expressed there, we could see that essentially most of the upregulated and especially most downregulated genes are found in angiosperms, with Markantia having a considerable number of genes both up and downregulated. This looking in general at all orthogroups. And if we break this down in conserved orthogroups or gained or lost, we essentially see the same, where plants such as Physcometrium or Selaginella actually don't respond very strongly to hypoxia. And I will come to this a bit late in my talk. But then looking deeper in the identity of these orthogroups, we looked at those that were at least upregulated or downregulated in 3 species. So which then would span in more than just one division. And here are the groups the functional groups we could divide them into. And if you look them in the, let's say, horizontal direction, where you see essentially an orange line would indicate conservation of the induction, or a green line would indicate conservation of repression, we can say that there is a stronger conservation among induced genes rather than the repressed ones. And then we can use this data set also to ask whether potentially these induced and repressed genes are potentially targets of the ERF7 transcription factors that I introduced before. So what we did here was using a resource produced by Mike Holdsworth. So this is a pentuple mutant that is lacking all 5 of the ERF7. And we exposed those plants to hypoxia and compared the response with the wild type. And we looked at that after 2 hours hypoxia and 10 hours hypoxia. And from this analysis, it was quite clear that, and here the darker color indicate the genes that rely on the ERF7 to be up or down regulated. So it's quite clear that the ERF7 are important to induce about 50% of the genes upregulated in the first 2 hours, but then the role seems to decrease with time. And the role in repressing genes is also quite milder. And if we then take these genes here that are relying on the ERF7 and we map them on the orthogroups we analyzed before, we can see that indeed most of the upregulated genes are controlled, at least in Arabidopsis, by the ERF7. And so we might assume that are also controlled by ERF7 in the other species we analyzed. And we did that later. But we also focused specifically on this group here, which includes the most conserved upregulated genes. And we looked at the identity of the orthogroups here. And we found that essentially we have exactly those genes that I mentioned before, those involved in glycolysis and fermentation, again, highlighting the importance of metabolic adaptation under a hypoxic condition. This then led to the questions, is apart from their identity and the fact that they respond to hypoxia, is also their regulation conserved or not? So are they targets of ERF7 in all the species we analyzed? So I'm showing here a sort of simplified version of the diversification of divisions in land plants. And I'm listing essentially the ERF7 and the different enzymes that catalyze their degradation in an oxygen-dependent manner. So what we notice is that, for example, in Arabidopsis, both the ERF7 and the PCO enzymes are relatively large gene families, while arginine transferases and ubiquitin ligases are more restricted in numbers. And from work also conducted before, we know that this is quite well conserved. Actually, this is even conserved in animals and in yeast, with the exception of PCOs that are only present in multicellular eukaryotes. If then we look at the substrates, and in particular the ERF7, we see that essentially we found at least one ERF7 in all vascular plants, but we couldn't find any ERF7 in non-vascular plants, in bryophytes. I define ERF7 as ERF transcription factors, which have a number of domains that distinguish this protein. What we found is, though, that with the exception of algae, in Marchantia and in Fiscomitrium, we could find some ERFs which have a cysteine in the second position. They're not just classified as ERF7. So we tried to characterize them a bit. So first, we looked at the conservation of the endagron portion in the ERF7 in vascular plants. Just using the DNA binding domain, we could recapitulate quite nicely the clustering of different species according to their divisions. And when looking at the endagron, if you remember, I presented before that this is extremely conserved, and indeed, with very few exceptions, we found this sequence, not just the cysteine being conserved in the second position, but this glycine, alanine, and isoleucine. When we look in gymnosperms, this is still very strongly conserved. And this conservation is sort of lost, but with variations in ferns and in lycophytes. So indicating that potentially there is a different interaction with the enzymes that we know are operating on this portion of the protein. So if we put together this information and we try to reconstitute the evolution of the ERF7, well, we know that ERF transcription factors are present in the whole plant kingdom. It's a plant-specific class of transcription factors. We can hypothesize that ERF7 as targets of the endagron pathway arose in the last common ancestors of all vascular plants. And then progressively, we observe a sort of specialization of the endagron that is quite fixed in angiosperms and instead apparently relatively variable in ferns. So the fact that they have this feature would indicate that they are substrate of the endagron pathway. So can we test this? This is what Laura did. Here we have again a sort of reminder of how their degradation is controlled in an oxygen-dependent manner. And what Laura did was taking one or two members of these ERFs, including those that are not ERF7, but are MC ERFs in bryophytes, and cloned them and put them in the ERF7 mutants that Mike generated, and then challenged these plants with well, either hypoxia or submergence to as a mean to remove oxygen, or using drugs that inhibit the proteasome, such as bortezomib. And what was clear is that in the bryophyte sequences are not stabilized under hypoxia, although they're also controlled by the proteasome. While instead, and we received quite some criticism by reviewers here, but definitely there is a stabilization of these transcription factors, these ERF7 transcription factors from different species. It's just that they're extremely labile, these proteins. So we will never see a strong upregulation as when we toxically inhibit the proteasome. Nonetheless, they do stabilize. What we also tested was if we get the same effect when we kill the proteasome by removing PRT6, the ubiquitin ligase. And this was a kind of unexpected result. For the non-angiosperm ERF7, we could see stabilization in PRT6, but we could also see that for the bryophyte ERFs, which were not controlled by oxygen. So this part is still a bit puzzling for us, as if they are substrates of the endogenous pathway, but they are not sensitive to hypoxia, potentially because of a strong interaction with the enzymes that modify them. So what about then these ERFs? Can they control the response to hypoxia? Here we had to take two different strategies. For plants that we can transform, such as Marcantia, it was relatively easy. We used CRISPR to knock out these genes. We obtained independent lines. Sort of unfortunately, they all had exactly the same mutation, but these were all originating from different insertions of Cas9, which eventually was segregated out, so was not particularly important for us. They definitely interrupted the protein at the level of the DNA binding domain, but definitely we didn't observe absence of activation of hypoxia responsive genes in Marcantia. If anything, this was even strongly enhanced. For species that for which instead we don't have a transformation protocol, such as lycophytes, then we couldn't knock out the gene in Selaginella. So what we did was cloning the promoter of hypoxia-inducible genes from lycophytes and co-transforming them in Arabidopsis protoplast together with the transcription factor from Selaginella. And what we observed is that in this case, they could transactivate, which is an indication for us that indeed probably this ability to regulate, for example, fermentation genes arose quite early in the last common ancestor of vascular plants. So what about these other MC ERFs that we find in bryophytes? Are they belonging to one single group, or are they more dispersed? Well, it's the second case. We find them in several different groups of the ERF family. So indicating that potentially this recruitment by the endogenous pathway, by the presence of a cysteine in the second position, is a common feature for ERFs. There is a tendency on the endogenous pathway to recruit these proteins. There was a very odd observation we had. So this is the group 7, the group 7 here, which includes all the Arabidopsis sequences, includes the Selaginella sequences, but there was also a Marcantia sequence here, which puzzled us because, well, first of all, this is not an MC protein, so we hadn't tested before. But looking at several Marcantia species transcriptomes, we could find it in most of them, which might be interpreted potentially as a sort of relic, as an ancient ERF7, which was not MC or has lost the MC feature, but maybe was present already before for lycophytes, and then lost, for example, in mosses. So this allows us to sort of update the tree that I mentioned before, with the fact that, as I mentioned, other ERFs, not ERF7, are apparently recruited by the underground pathway from different groups on different divisions. And the fact that potentially we found this ERF7 in liverworts that might anticipate the presence of an ERF7 ancestors in plants. So bringing all this together, my first series of take-home messages is that, well, first of all, the set of enzymatic reactions needed to control proteins in an oxygen-dependent manner is present in all land plants. The ERF7 apparently were recruited as a substrate together with the evolution of vascular plants and potentially roots. But also that the underground pathway tends to recruit ERFs in other groups of plants. And at the level of responses that are generated, although potentially the regulation by ERF7 is not so much conserved, those responses that involve metabolic adjustments were already present in the last common ancestor of all land plants, and fermentation is one of these features. So this is what pertains the non-vascular plants and non-flowering plants. But then when I'm presenting this, and when I was presenting the mechanism for oxygen sensing, I also often get the question, what happens with plants that then normally cope with hypoxia because they're aquatic, for example, they're constantly submerged. And this is actually work that was carried out by a former PhD student from here, from Toulouse, Jimena Chirinos, a great postdoc now in my group, with the help of Vinay Shukla, also postdoc in my group. And together they tried to address this question as to whether the habitat in which plants or which plants colonized in some way affected the way oxygen sensing is organized. And for this, Jimena and Vinay focused on 3 different habitats characterized potentially by different exposure to hypoxia. So first, they looked at plants that are very unlikely to be submerged, plants that live on trees like epiphytes. They also looked at xerophytes, plants that grow in very arid conditions, so are very unlikely to be submerged. And finally, they used macrophytes, plants that grow in a perfectly aquatic environment. Often they live on the surface, but at certain stages of their life, they're actually submerged. And Jimena had a survey on essentially all plant species available for which a genome was sequenced and which could be assigned to one of these 3 habitats. She also used a panel for sort of control plants, mainly crops. And so this is the result of her analysis, where she looked essentially at the conservation of the enzymes that participate to the underground pathway here, and the substrate of this pathway, the ERF7, the little zipper2, and VERN2. And what was quite obviously striking to her eyes was first the fact that aquatic monocots here, seagrasses, lost apparently don't have one type of PCO enzymes. These PCO enzymes are those that are induced by hypoxia, to create, that are constitutively expressed hypoxia. They're sort of the repressors of hypoxia response under aerobic conditions. So it would kind of make sense that they are not present anymore. And also the fact that this group of substrates, like the little zippers, was clearly not present in these genomes. So this is first observation. The second, compressing this analysis and just looking at the ERF7, it's quite obvious that there was an expansion of ERF7 in general or in species that are associated with an aquatic habitat. And Jimena expressed it as a relation between the number of ERF7 and PCOs, which was quite clearly higher in macrophytes. Looking more specifically at, the other way we can observe the same is just looking, just using all these features here, or all the different endogram pathways and substrates, and running a PCA and seeing that essentially macrophytes are those that separate the most, and the separation is explained mostly by the number of ERF7 and secondarily by the number of PCOs. But also looking more specifically at the endagron of this ERF7, what Jimena noticed is that she could identify some kind of habitat-specific variations in the endagron. Some of them quite early in the sequence and some later. So she focused on this, labeled in pink. And she looked at what could have been the consequence of this variation. We are also testing this experimentally, but the experiment is running right now, so we don't have the answer yet. But the prediction is that, so if we try to model and dock this endagron, this portion of the protein, into the catalytic site of the cysteine dioxygenase, the PCO, and here we see the classical pair from Arabidopsis, PCO4, our favorite PCO, and the one for which a structure was solved, and RAP2.12, sort of our favorite ERF7. We see that quite clearly the peptide can enter inside this pocket. But these modified endagrons instead acquire structure. They have an alpha helix that potentially, or at least in the docking simulation, would prevent it from entering inside the catalytic side of the enzyme. So potentially this might indicate that this ERF7 escaped the regulation by the endagron pathway. We are testing this at the moment using an heterologous system. You might wonder if this is biased because this is modeled on the structure that we have on PCO4. So what Jimena did was also looking at the same, predicting the structure of the PCO in the aquatic species, and essentially the prediction is the same. Those endagrons that have a structure can't enter inside. So it seems that indeed there is an effect of habitat colonization on differentiating numbers and structures or sequences of this component of the endagron pathway. Of course, there could be another explanation for this diversification, which is the fact that these species have very different anatomies. And so if you remember, I mentioned that their meristems, the meristems in Arabidopsis tend to be hypoxic. So it might be that there is also a difference in the physiological hypoxia that is experienced normally by this species. So Jimena undertook a quite challenging task of measuring oxygen levels in the meristems of two species of macrophytes, xerophytes, and epiphytes. where she observed quite some variation in the oxygen levels that are experienced by meristems and the steepness of these oxygen gradients, but essentially she didn't observe any shared pattern across habitats, so indicating that this probably is very anatomy dependent and species dependent. So we kind of ruled out for the moment the possibility that there is a correlation because of anatomies, and we expect that this is more driven by the environment. We also had a look at then the response itself. So these differences in ERF7 might translate in a different type of response. So again, Jimena chose at least 2 species from these 3 different abstracts habitats, and she again exposed them to hypoxia and then extracted RNA to measure gene expression. And running orthogroup analysis, what she noticed is that maybe unsurprisingly, macrophytes are those species where the least number of genes is changed, and also the magnitude of change is reduced across all the species analyzed. And I say that this is potentially not unexpected because, of course, these plants are constantly sort of exposed to hypoxia, so they might be sort of primed or used to such conditions. On top of that, she then looked at potential conservation in these responses. She's still currently analyzing them. She sees mainly for xerophyte quite specific high number of orthogroups being up and down regulated. But for example, she couldn't see any conservation in the downregulation in macrophytes. And interesting for us was this group here shown in red, which essentially brings together a group of very conserved genes or orthogroups induced by hypoxia. And interestingly, we couldn't find the equivalent among the downregulated. So suggesting that the conservation, again, is more at the level of induction, while the downregulation of genes is probably more species-specific. And in this group, group of 11 orthogroups, we find, again, the genes that are involved in metabolism and fermentation, and also the PCOs. But also a number of transcription factors, some of which we know already about, the ERF7 themselves. So this is probably a sort of feedback loop, but also new transcription factors that either were known but not so much studied, or never identified as a relevant component to the hypoxia response. And we are currently actually making knockouts of these genes in Arabidopsis. Again, they belong to gene families, so we need to wait to have double and triple mutants. But what we looked for the moment was the cis regulatory elements that are recognized by these transcription factors because they have been characterized. And again, we see a significant overrepresentation of hypoxia-responsive elements, specifically in upregulated genes, so indicating again that probably ERF7 are positive regulators of gene expression. And another interesting feature for us is the distribution of these motifs. With very conserved and completely independent from the habitats positioning of these DNA elements, which sort of suggests a specific architecture on promoters that are meant to respond to hypoxia. And with this, actually, I'm concluding with the second series of main messages, where I would like you to remember that essentially we identify an increased number of ERF7 and variation of the sequence and dendron, which sort of correlates with underwater growth. The fact that probably this diversity doesn't correlate with anatomies that restrict oxygen levels at the level of the shoot apical meristem. and the fact that aquatic plants tend to respond less or less strongly to hypoxia challenges. And that rather we observe a conservation in induced genes rather than in repression, where accommodation of survival is probably quite species specific. And so finally, before taking your questions, if there is any, I would like to thank all the people involved in these two projects, my group, and of course, all the funders that supported this work. And thank you for your attention.